In corner one, we have Lian-Huan, the adventurer Wei. Lian hails from China, where she studied chemical biology. Lian-Huan loves to explore nature and isn't scared of anything, from scuba diving deep in the ocean to ice climbing up high in snowy mountains. She's seen it all. However, there is one thing that maybe can scare Lian-Huan away from her mission. She will close her eyes when she's forced to watch scary movies, but we don't blame you. And once again, representing neuroscience, we have Lian-Huan. In the chat, please give it up for Lian and representing neuroscience. And in the second corner, we have Noreen, the cookie monster, Gentry. Noreen hails from Iowa and studied chemistry at Iowa State. Noreen is the biggest basketball fan, and she even once camped in an ice storm to catch a game. Coming in as a close second to her love of basketball is her dedication to memes. And make sure you get ready for her talk because she's going to have a couple memes thrown in there for you guys. Her favorite cookie is a hot and fresh peanut butter chocolate chip, and she will eat them every day to stay strong. So also give us a shout out for other peanut butter chocolate chip cookie fans in the chat as well. And once again, representing chemistry, we have Noreen. Okay, and so the way we decide who goes first around here is with a coin flip. So Heather, can you bring up the coin onto the screen? And in the meantime, while our coin is getting brought up, let's see. Lian-Huan, would you like to call heads or tails for us?
Yes.
Okay, let's see what it says. Ooh, heads. Okay, so Noreen, I guess that means you get to choose.
I'll go second.
Okay. And before we get started here, I want to make sure this is a clean fight, but do you have anything you want to say to each other just to ruffle some feathers up a little bit?
I've heard you're not afraid of anything, but you should definitely be afraid of losing to me.
Ooh, okay.
Yeah, I heard you gonna talk about nanoparticles. Maybe you're gonna get nano score.
Ooh, a nano score. Okay. Well, let's battle this out with some science to decide who's on top. And Lian-Huan, you can go first. So I'm excited to hear your talk.
Okay. Can you guys see my PowerPoint?
Yes.
Yeah, it looks great.
Okay. Hi, everyone. It's a great pleasure to be here. My name is Lian-Huan Wei, and I'm a postdoc in the Department of Neuroscience at Yale. Today, I will show you how I hunt for ALS genes with molecular scissors. Okay, a brief introduction again about myself. So I come from China. I came to the US at the end of 2018. I like trying cool activities such as skydiving, scuba diving, ice climbing, hiking, camping, and skiing. I like to be a scientist because I can learn new things every day. Okay, let's start our science journey. First, I need to tell you what is ALS. Have you guys ever heard of the Ice Bucket Challenge? It went viral in 2014. This activity was to raise money and awareness for the ALS disease. Some well-known people have been diagnosed with ALS, including Stephen Hawking and the famous New York Yankees baseball player Lou Gehrig. ALS disease is also called Lou Gehrig's disease. Specifically, the ALS is the acronym for amyotrophic lateral sclerosis. We have neurons in the brain and the spinal cord that control the muscle movement. In ALS patient, these neurons are damaged, so the brain was not able to control the muscle movement. People with ALS will gradually lose the ability to speak, eat, move, or even breathe. There is no cure for ALS at now. What we want to know is what's happening in ALS patients in the molecular level. To address this question, we need some background. You might already know how the cells make protein. This procedure is also called translation. So the protein making information are stored in DNA. DNA can be copied into pre-mRNA, which has exons and introns. The introns will be removed out, and only exons are returned in the mature mRNA. Then the protein-making machinery will translate the protein from the translation start site to the translation stop site. This is what usually happens in the cells. However, in the ALS patients, something unusual happens. So there is one ALS gene. It has hundreds to thousands of GGGGCC repeat sequence in the first intron. When the cells make mature mRNA, this giant intron with the repeats will return in the mature mRNA. And somehow this giant intron with repeats can trick the protein machinery to make protein from here instead of from the start site, translation start site. And unfortunately, these proteins made here are toxic. The protein will accumulate in the neurons in ALS patients. As you can see here, the protein aggregates here, they will kill the neurons at last. So the scientific question we want to ask here is, why does the abnormal translation happen? And can we find the cell factors that help or prevent the abnormal translation? So to answer this big question, we need several steps. First, how do we monitor this abnormal translation? Well, the answer is we can make a reporter for it. We can take out this fragment with the repeats and attach a green fluorescent protein gene after it. When the cell translates this fragment, it also translates a glowing protein. So at last, we have a group of bright cells that indicate this abnormal translation. As you can see here, these bright cells beautiful under the microscope. So now we have a reporter for this abnormal translation. Then we're gonna ask, how do we know which gene affects abnormal translation? Well, we have a strategy. We can disrupt each gene in the cell to check the cell brightness. So remember, we have a reporter that indicates the abnormal translation through the cell brightness. If we disrupt a gene and the cell brightness changes, that means this gene affects the abnormal translation, right? But how do we disrupt genes in the cells? Now it's time for our star to show up. Welcome our star, CRISPR-Cas9. Yeah, it's the molecular scissor. You might know that the 2020 Nobel Prize for Chemistry was awarded to Emmanuel Charpentier and Jennifer Doudna for their discovery of this molecular scissor, CRISPR-Cas9. And this molecular scissor has two parts. One is a Cas9 enzyme. It can cut the gene. And a second part of the molecular scissor is the guide RNA. It tells the Cas9 enzyme which gene to destroy. So next, we can design guide RNAs for all the genes in human cells. After combined with the Cas9 enzyme, we can send an army to the cells. And each soldier is responsible to kill one individual gene. Okay, so this is our principle of the experiment design. So when we kill gene A, the cells get dimmer. And when we kill gene B, the cell get brighter. Oh, sorry. Almost tell you the answer. Can anyone in the audience tell me? Oh, okay. Okay, now we know that if we kill gene A, the cells get dimmer. That means gene A has abnormal translation. So anyone know if we kill gene B, the cells get brighter? What does that mean? Yeah, it means this gene B gonna prevent the abnormal translation. So now we know that how to destroy a gene in the cell and how to identify genes that helps or prevent the abnormal translation. Next, we need to solve this question. How can we check the brightness of millions of cells? You might say that we can check the cells under the microscope. But do you know how many cells we have? It's about 20,000. Actually, we have multiple guide RNAs for each gene, so we need to check the brightness for millions of cells. How do we check the brightness for millions of cells? Well, actually, we have a very cool instrument called the fluorescent activated cell sorting, FACS for short. So this is the FACS instrument has a laser beam. It can activate the brightness of the cells, and it also has a Shell's fluid that can help cells line up to go through the detector one by one. So then we can separate the cells according to their color or the brightness. And what even more amazing about this fact is that it can detect the cells by a speed of tens of thousands cells per second. One second for over thousands of cells. So we detect the brightness of millions of cells within a few hours is not a big problem. Okay, now we know how to check the brightness of the cells. Then we come to the last question, like check the loss of which gene gonna cause the change of a cell brightness. So we have a routine biological method that we can take the guide RNA out from the cells, so we can know that which genes or which guide RNA decrease the cell brightness or increase the cell brightness. Then we can know that which gene help or prevent the abnormal translation. So just give you a sense how many cells we need for one screen. So this plate has one plate has about 20 million cells. We need over 50 plates for one screen. So we need a lot of cells for this experiment. And this is typical data we got from the screen. The x-axis represents the brightness, and the y-axis represents the cell number. So after treatment of the cells with the molecular scissors, some cells get dimmer and the other cells goes brighter. So the dimmer cells locate on the left side of the histogram, and the bright cells locate on the right side of the histogram. And we take 10% of the dim cell and 10% from the bright cell. And after take out of the guide RNA, we should know which genes are the helper or the preventer of this ALS abnormal translation. So in summary, to study the abnormal translation for ALS or some other biological event, we can make a report for it. Then we can send an army of CRISPR-Cas9 to the cells and kill each individual genes. Then we separate the cells according to their brightness. At last, we separate the guide RNAs from the selected cells and identify the loss of which gene led to the change of the cell brightness. After we know which genes are the helper of the ALS or the preventer of the ALS, we can develop drugs. We can then use the drugs to inhibit the ALS helper gene and activate the ALS preventer gene. That's how we help treating the ALS disease. That's all my talk today. With that, I want to thank all my members, all the members in my lab, in our lab, especially my mentor, Junjie Guo. He's always supporting me. Yeah, thank you so much. I'll take questions. Okay.
Hi, Lian-Huan. That was such a great talk.
Thank you.
It's also so cool to see, like, you know, the Nobel Prize work. You know, I think the students, we talked about the Nobel Prizes when they came out, but it's cool to actually see them, you know, being used. Yeah. Okay. And I also think all the students had a great time hearing the talk as well. And we will collect all of everyone's questions. If you have any other additional questions for Lian-Huan, please put them in the Q&A section. And after both talks, we'll ask you all of them together.
Okay,
yes. But in the meantime, we're about to give all of these students a poll. And this is a poll for you to determine how well Lian-Huan's talk was. And the poll, which you're going to get in just a moment, is going to have 3 questions. The first question is, how understandable was the presentation? Were you able to follow most of what the presenter was saying? And this is going to be rated on a scale of 1 to 5, with 1 being you were completely lost and 5 being that you understood everything, or anywhere in between. Question 2 is going to be, how much did this presentation spark your curiosity and your imagination? Even if you don't want to do the research yourself, like, how much did it make you think, wow, that's actually pretty cool? And that's also going to be a scale of 1 to 5. And the last question is, how much more do you feel like you know about this topic compared to before? And that's also going to be on a scale of 1 to 5. So you should be getting the polls right about now. And if you've already finished filling out the poll, you know, think about any questions you have for Lian-Huan so we can ask her at the end.
Today I'm going to talk to you, oops, spoiled my joke. Getting my laser pointer up really quick. Perfect. Today I'm going to talk to you about using nanoparticles to help harvest renewable energy. And since it's May 4th, here you go, May the 4th for all the Star Wars fans out there. But before we get into the science part, I wanted to talk to you a little bit about my journey to science. So I originally grew up in Iowa, this nice red blob right here in the middle of the country, a little ways away from Connecticut. And I didn't venture too far when I went to undergrad. So I majored in chemistry at Iowa State, which is right here in the middle of the state, which I still think is kind of funny that I decided to major in chemistry because growing up, I really didn't enjoy science that much. I thought it was a little bit boring and that everything that I learned in books was already understood. So I didn't really understand what there was to study. But at college, I gained a better appreciation for everything that we don't know. And that the things that we don't know are really exciting because we can use science to try to answer these questions. And as we learn more and more, then there's more questions for us to try to answer. So I've been at Yale for the last 4 years doing a PhD in chemistry, but chemistry has also paid for me to go to a lot of cool places where I've met a lot of really interesting people. So you never know exactly where science will take you, both intellectually and what space you'll end up going to. So in thinking about science as a way to solve problems, one huge problem that we have right now is that the world uses a lot of energy and the population of the world is continuing to grow. So here's this huge table that I'm going to walk you through each and every single number. So buckle up. No, I'm just kidding. Here's your first meme. Looking at a table like this is kind of hard to understand what's going on. So instead of talking through all of these numbers, I want to zoom in a little bit. Instead of thinking about the energy that the world uses, I'm going to ask you a simpler question. What energy did you use today? What are certain things that you did? Feel free to throw it in the chat. Light energy, your body, same, lots of laptop battery, yeah. That was one of the things that I was thinking about. Right now, all of us have some amount of energy that we're using for our computers. Maybe you charged your computer before this talk. Maybe you're using electricity right now. Do fun things like watching YouTube and TikTok. I know my lights are turned on right now. That takes energy. Heat energy, yep, you need that to make chocolate chip cookies. And even things like the shirt that you're wearing, that takes energy to get those materials, to make the t-shirt, then for you to go to the store and purchase it or for it to get delivered. All of these things add up. And when you think about your entire day, everything takes energy. So to meet all of these energetic needs, I want you to think about a future day, hopefully not too far in the future, where we can use renewable energy sources to meet all of this, meet all of our energy needs. So think about wind, think about solar. What might that look like? Think about what did you do today to make sure that when you plugged in your computer that you would get energy? I know I didn't think about it at all. I plugged in my computer and I assumed that it would work, and it worked. So for most of us, using renewable energy sources actually probably won't look that much different than what we do now. But for the people that make sure that this supply of energy meets the demand of energy, things might be a little bit different. So can anyone tell me what some of the considerations are when we use renewable energy sources? Why it's sometimes kind of tricky? Timing and location, yeah. Supply and demand, land, environment. Yeah, and if we think about using something like solar power, there are certain days when it's not sunny, where it's rainy, at nighttime, we don't have access to the sun. And look at all of this energy that we use at night in order to keep our lights on, in order to watch TV. All of these things take energy. So in order to use renewable resources, we need to find ways to store the energy, to store the sun's energy when it's sunny out so that we can use it at night. So I think when I think about storing energy, this is an example that comes to mind. So imagine that you're at the bottom of a hill with a bucket of water, because why not? If you take this bucket of water and you walk it up the hill, you can see that as you walk up the hill, my line here gets taller and taller until you get to the top of the hill. And at the top of the hill, when you're not moving anymore, you don't have any kinetic energy. You don't have any movement. But you do have a lot of potential energy that you've stored up now. So potential energy is the potential to, you can think about this as the potential to do kinetic energy. And that's easier to understand if we think about taking your bucket of water and dumping it back down the hill. So when you take this bucket of water that has all of this potential energy stored up and dump it down the hill, then this water will move. And we can take advantage of that kinetic energy by putting something like a water mill at the bottom of the hill, that as this water rolls down the hill, it can turn this wheel. And as this wheel turns, we can use that to generate electricity. Yeah, exactly. Kinetic energy is moving energy. Perfect. So this is actually a strategy that we currently use to store energy. So one way that we do this is when it's really sunny out and we have way more energy than what we could ever want. We take the sun's energy and we do something that's really hard. We pump water up a hill. That's something that I wouldn't want to do, so I'm really glad that we have technology that will carry the water up the hill for us. So it might look something like this. We'll pump this water up the hill into this reservoir. So that then when it's at nighttime and we don't have access to the sun, your house lights might be turned off. We can take the water where it's stored up all of this potential energy and we can flow it back down the hill. And as we flow it back down the hill, we can spin this wheel and generate electricity. And using that electricity, we can power our house. So that's one way that we can store energy. And as you think about energy, I think it's important to remember the conservation of energy. So energy is not created or destroyed, it simply changes forms. So we have this back and forth between kinetic energy and potential energy, and we constantly go back and forth. So this is one example of how we can store renewable energy, but there's other ways that we can store renewable energy and other approaches that we can take. So one that you might be familiar with is batteries. And the example that I'm going to talk to you about today is storing energy in chemical bonds. And since there's a lot of different needs that we have for energy, there's a lot of different locations and places, we really will need a wide variety of ways to store renewables. So this is just the example that I'm going to talk to you about today. So if we're thinking about storing energy in chemical bonds, I think first it's important to talk about what a chemical bond is. So I'm going to give you a simple definition. Chemical bond is when we stick two atoms together. So here's an example of hydrogen, which is one of the lightest atom, and we're sticking hydrogen together using electrons or negative charges that act as glue. And in this case, this hydrogen is really happy. It's in an energetically favorable position, which I'll talk about as happiness. And it takes a lot of energy to break a happy bond. But in order to store energy, we need to think about breaking and making chemical bonds. So the fact that it takes energy to break a happy bond like hydrogen seems like maybe there's a little bit of a problem. So to address this problem, I use a nice tool. I use nanoparticles, which will help me make and break bonds. And of course, when you put nano in front of everything, it definitely sounds scientific, but what does it mean? So what is a nanoparticle? The way that I break it apart to think about is you can think about this as a particle. So in this sense, I'm talking about a small cluster of atoms that's nano, super, super small. But I still think that's a little bit challenging to fully grasp what super small means. So I'm going to walk you through how to think about atoms in comparison to nanoparticles in terms of items that we already know. So I know atoms are super, super tiny. We can't see them with our eyes, but I want you to scale up and pretend like one atom is actually the size of a marble. If I take a bunch of marbles, I can add them together until I make a sphere roughly the size of a basketball. So it might look something like this. And this basketball made out of marbles would be an example of a nanoparticle. And to give you a sense of how tiny atoms and nanoparticles are, if we were to take this example and use an actual basketball, can you guess how big it would be? Yeah, it'd be very big. It'd be roughly 1.7 times the size of the Earth. So think about a basketball that you can hold in your hand. That's the difference in size from basketballs to atoms and nanoparticles. So it's pretty crazy. So when I talk about nanoparticles, I want you to have in the back of your mind this basketball made out of marbles. So how can I use that basketball made out of marbles to help me make and break chemical bonds, which will help me store energy? So hydrogen is a happy molecule, and it takes energy to break this happy bond. So the way that I like to think about this is with something that I can relate to. So let's think through an example. Suppose that you are sitting on your couch. This, for me at least, is a very happy position. I don't want to move. There's not much motivation for me to get up and go do something else. But let's say maybe your friends text you and say, do you want to go outside and hang out, maybe eat some cookies? Because of course, cookies are the best. You know that this difference in happiness, so if we look at our scale, you'd be this happy on your couch, but you'd be even happier out with your friends. You think, yeah, that sounds great. But then you start to think about all the steps that you have to take to go hang out with your friends. You have to get up off your couch, exhausting. You have to go around your house and find your sweater. You have to find your shoes. That just seems like a lot of work. So as you start to think about this barrier, you decide, maybe not. Maybe I'll just hang out on my couch. But we can use something like a catalyst. So suppose that you have someone in your household, like your parents or a sibling, that brings you your sweatshirt and brings you your shoes. That lowers the barrier for you getting up to go out and hang out with your friends. And it makes it easier for you to go do that. So similarly, molecules also have unhappy intermediate steps. If we think about our happy molecule, there is another molecule that would be happier, that would be lower in energy. But in order to get there, we have to go through this unfavorable intermediate step. We have to get off the couch. So we can use something like a nanoparticle, which nanoparticles can do a lot of different things. Maybe they'll bring electrons, which will act as the glue to stick these atoms together. Or maybe they'll bring atoms to these other atoms so we can stick them together. These nanoparticles help to make things easier. So far I've talked a lot about the happiness of these molecules in the initial state and in the final state. But it's important for scientists to really understand how this catalyst works so that then we can lower this barrier and make it even easier to store energy in chemical bonds. So we really want to understand how this process works. So in my research, I try to understand these nanoparticles. And one of the types of nanoparticles that I study is titanium dioxide, which I have a picture here of titanium dioxide mixed in water. And titanium dioxide, or TiO2, is in sunscreen, and it's even in powdered sugar donuts, which is pretty wild. So in my studies, I take these solutions and I add electrons to them, which looks something like this. And what's really cool is I can see a visible color change. So when I add electrons to my solutions, they turn this nice blue color. But as we're thinking about using these nanoparticles to help make and break bonds, we want to use those electrons to help us store energy and to help us make bonds. So we can take these electrons and make bonds, and we can do that a bunch of times. And through this process, then I see my color go from blue back to clear. So as you're thinking about this process, you can think about energy storage with all of these molecules that we can make. And then this catalyst at the end is ready to be regenerated to help us make more bonds. All right, so in summary, some of the things that you can remember from this talk. Catalysts are super helpful because they lower the barrier to making and breaking chemical bonds. So you can think about this next time you're on the couch and someone helps motivate you to get off the couch and go hang out with your friends. You can thank them for being a great catalyst. A nanoparticle is like a basketball made out of marbles. So you can have that vision in your head next time you hear about nanoparticles. And nanoparticles help us store energy in chemical bonds. So as you're thinking forward, I want you to picture a day to think about what these nanoparticles can do to help us use renewable energy. So I wanted to give a shout out to all of the people in my lab that I've worked with in the past 4 years. I've had a lot of fun, and to all of my funding sources that have paid me to do all of this fun science.
Thank you so much, Noreen, for such a wonderful presentation. I think the students in the chat are a little bit sad about the donuts, though. They don't know if they want to be eating titanium with their donuts. They thought it was just sugar. But other than that, they seem to really enjoy your presentation. And now you guys can tell Noreen how much you liked your presentation with the poll. So it's going to be the same poll as last time, but this time you're going to be answering the questions for Noreen. And the poll should be coming up on your screens now.
The students have asked lots of fantastic questions. And so I'm going to pose some of them to both of you. So first I'm going to start with the neuroscience questions. And so one question about ALS, is ALS a genetic disease, meaning does it come from our DNA, or is it based in some other method? How does a person get ALS?
The ALS can be a genetic disease, but also some people get ALS. They call it sporadic cases. So for that cases, we don't know why it happens. For some patients, ALS patients, about only like 5 to 10%, very small fraction of the people are genetic. So they get the disease from their parent or their grandparents, but for other people it's sporadic. So it's still a big mystery. Yes, it needs a lot of study.
Well, that's exciting to know, because that means that our students still have a lot of interesting questions to answer in the future. Okay, next question is for Noreen. Noreen, how does that wheel turn into electrical energy? Is mechanical energy being turned into potential energy? Can you explain the wheel analogy a little bit more?
Yeah, that's a good question. I don't know exactly all of the specifics on how those systems work, but I know the general principle is, especially as you're thinking about putting the water down the hill, is that you take that kinetic energy, that energy of movement, and you use it to turn something else. And as that turns, then we can take that and turn it into electricity. But that's a really good question that I don't know all of the specifics, the specific mechanisms.
Again, another great opportunity for our students to research on their own. That's incredible. Okay, so another question about neuroscience. Let's see, how many genes do you have to test, Lian?
About 20,000. So you mean how many genes are needed to test to find the genes are responsible for ALS disease, right? Yeah,
Yes.
we need to test all the genes in our cells.
Gotcha. That's very interesting. There's so many genes in the human body, and having to go through all of them must be a very rigorous process.
Yeah, we need a lot of cells. Yeah.
Definitely.
Yeah, and high throughput procedure.
Yes. Okay, next question about energy and the nanoparticles. So how exactly do nanoparticles know which atoms to bring where?
Yeah, that's a really good question. I think that's something that scientists are really trying to understand. So one thing that's really interesting with nanoparticles is that I didn't have a chance to get into is that they have a large number of atoms that are at the surface of them. So if you think about your basketball made out of marbles, there's a lot of marbles that are close to the edge that you can actually touch, but there's some of them that are on the inside of the basketball that you can't reach. So the fact that there's so many around the edge leads to their ability to help us do different types of reactions. But there's different areas of the nanoparticle that sometimes are more reactive than others. And that's a question that a lot of scientists are trying to answer.
Thanks so much. Okay, back to ALS. So Lian, you talked about ALS helpers. Are ALS helpers good or bad? Do they help to prevent ALS or to facilitate ALS spreading?
Oh, sorry for the confusion. ALS helper, I mean, it helps the ALS happens, so it's bad. Yeah. Yeah, I mean the factors that like cause the ALS disease, so we don't want it. Yeah, it's bad. Yeah.
Okay, awesome. Okay, another energy question. So what would happen to the color change if you added protons or even neutrons into the titanium dioxide?
Yeah. So this is another thing that I didn't have a chance to get into too much, but actually a big part of the research that I do is thinking about not just the electrons on the nanoparticles, but also protons. So that's a great catch. We've actually found that, well, it's not super favorable to add a ton of negative charge to the nanoparticles. It's easier to add a negative charge and a positive charge, so a proton and an electron. So you get a net neutral. And then you can add more of these charges onto the nanoparticles and make more bonds, which then you can think about as storing more energy.
Great. Okay, for our last two questions, the students were also curious about your science journeys. So first for Lian, can you just tell very quickly about, like, you talked about you went on some adventures before you started your science. Can you just talk a little bit about that?
About my adventure?
Your adventures and how it led you into science.
Oh, yeah, I think I'm a very curious person. So I love the nature, also the life. I want to explore the things happens in the cells. So and I think like one day if I have a job, I want to experience new things every day. Like I kind of don't want a job that I need to repeatedly do it. Like it couldn't give me the feel of like refreshment every day. Yeah. So I study chemical biology from college, then also a PhD on the same major. Then now I come to the US, do a postdoc, like in similar area. So I think I started my science journey like from college. It's a long journey now. I love it. Many people think science is very hard, but to my experience, like anything can be easier if you stick to it. Like maybe I'm not the smartest person in my class. I'm not always on the top on my class, but I like it and I stick to it for many years. I invest all my time onto it, and then it led me to here. So yeah, I think, yeah.
Perfect. That's perfect. Thank you so much for sharing. Okay, Noreen, you're up. What was your science journey from, I guess, high school till now?
Yeah. So like I mentioned earlier, I didn't love science that much when I was taking it in high school. I thought it was kind of boring, but I did take a chemistry class my junior year of high school, and a lot of the class was struggling through trying to figure out what everything meant. And at that point, I grew to like the challenge, I guess, of trying to figure out what the textbook was saying, and to try to explain it to my friends. And I thought figuring that part out was really interesting. So I took a shot and went to college and declared my major as chemistry, thinking that I would teach. But at college, that's where I grew to realize that science isn't just what's in the textbooks, but that science is so much more than that, and that there's so many questions left for us to answer, and that we can use science to really start to ask questions and find answers for them. So in college, then I got into research, which I had no idea what research was. Really, it was a stepwise process of not really knowing what I was doing, but asking a lot of questions, talking to professors and to my other friends in chemistry. And through that process, I gained a better appreciation for what science was, what types of questions we can ask. And along the way, I fell in love with research and decided that I wanted to keep going. So regardless of what your background is, you can do it, and just try new things, ask a lot of questions.
Yeah, that's great advice. Both of you gave really inspiring stories, and I just hope everyone, all the students, know that science is accessible to all of you guys, and we're super excited that all of you keep coming back to learn more and more about science every week. Okay, so I'm going to pass it back over to Josie so that we can find out the results.
All right. Is everyone excited? This is the best part is the talks, right? But everyone hopefully is also excited to figure out who won. So I'm going to present to you all the results of your voting. As always, we appreciate both of the talks a whole lot. And we really appreciate all of the feedback. So I think the presenters come here because they want to present to you and also to work on their science communication skills. So all of the feedback that you have is really helpful for them as well. So can we get a drum roll going? All right, ready? So the first question we asked you all was, how understandable was the presentation? We asked you to rank this on the scale of 1 to 5. And you guys gave Noreen like almost a 4.2. So that's like very understandable. That's good. And then Lian-Huan's like right under 4. So that's pretty good. Most of you understood what she was saying. So that's fantastic. All right. Then for the next question, how much did this presentation spark your curiosity and imagination? For this question, you all gave Noreen like right about a 4. So a lot of people are very interested. And Lian-Huan is a little bit higher. Maybe it's called at 4.1. So still super close, but seems like we have some neuroscience futures maybe in this crowd. Very exciting. And then the last question we asked was, how much more do you feel like you know about this topic compared to before? For this, Noreen, almost a 4.2 there, and Lian-Huan like a 4.3. So you guys learned a whole lot in both of these talks. Absolutely fantastic. So now we're going to average all of these scores together to get us a final winner. Noreen comes in with an average of 4.1, also a 4.1. So everyone here knows that it's not usually a tie, but this time it's a tie. So we have no winner for you this week, but I think this just goes to show how much they loved both of your talks. So thank you both so much. A big congratulations to both of you, and thanks for all of the work that you put into it. Some people think that there should be a tie-breaking poll, but I think we're just going to call this 2 winners for tonight. So thanks so much.